Yes, good evening to all the participants. Welcome to the day 4 session on biological databases. So, today, majorly, we discuss about metabolic pathway, motif, and domain databases. So, before going to today's session contents, can we recap the last session contents? Yeah, let me take you to the chat box. Yeah. So, can we discuss the last session topics? Good. So, Akshata is already ready. Cake. We discussed about cake pathways, reactions, gene cards, bioinformatics database formats, brief about techniques for 3D structures. Good. So, anything else? Did she miss something? If she missed something, you can mention the topics. So, other than whatever she has mentioned, if something is missed, you can type, others can interact. Yeah, participants, from the beginning, I'm mentioning you that interaction is very important. Only through the interaction, I can come to know how enthusiastic you are, and even I can teach you more concepts. Or else, I have to limit because I have to. I'm thinking like you can't able to grasp so many concepts on a single day. Whether I have to teach more or less, that depends on your interaction. Any other topics? I mean, not last, last session topics. Biovia, good. So you came with a software called Biovia Discovery Studio Visualizer that I showed you how to open the 3D structure of a protein, how to visualize in the form of a good background. Okay, so we have discussed all this. And what else? Like what you feel something new on last day session, last session. Did you find something new? You learned something new on yesterday. I have explained you on data file formats, isn't it? So I explained about how the FASTA format looks like, how the GenPep format looks like, and can you tell me what are the other formats, database formats? What are the other database formats I have explained you other than FASTA and GenPep or GenBank? Good. So we also defined something about refinement resolution for a 3D structure. Good. So very good. Once again from Akshita. XML. XML is extensible markup language. This is a database language. Okay, so we have discussed about that. Good. So from even from Irfan, came to know structured data format. We have discussed about this. Like whenever you want to work on more docking simulation, I told you to download a single chemical molecule or multiple chemical molecules in the form of data set. So what format you can use even if you want to download the form of a data set, you can use STF format, structured data format from NCBI PubChem. And I explained you about another database called KEGG drug, KEGG. I think she has explained, but okay. KEGG also explained about drug, isn't it? So what format we can download for a drug molecule in KEGG? Whether is it possible for us to download in STF format? No, it's not possible. Then what format we can able to download? It's not PDB. Of course, we have discussed more about the PDB, how to edit the coordinates, how to visualize it. It's not even mol2. Right well, but it's not mol2. What is the exact format? Anyone can 3D? Yes, of course, it's a 3D format. What is that 3D format? It's not PDB, it's not mol2. So you have to watch, okay, even you can watch once more. Okay, we already received the recorded version. Can I mention the correct answer? MOL. MOL2 is different format. MOL is different format. SDF is different. SD is different. Okay, so there are different formats. We also have SD. We also have SDF. We also have MOL2, MOL. Okay, there is a software called Open Babel. Now I can show you. Good kind of interaction I found even in this session. Let me take you to the today's session. So there is one software called Open Babel. Okay, in Open Babel, this is a chem informatics software. If you want to convert something like mol to mol 2 or mol to PDB, you can use this. Okay, so Open Babel is an online software. Okay, you have to install, and after installation, you can work it on online. But there is also a software called Babelwin. For this, there is no need of first you have to download and install. After installation, there is no need of internet. Without internet, if you have a format of mol, you can convert it to mol 2. Okay, so Babelwin is one of the file conversion format. I think this is not the exact website. Babelwin file conversion software. File converter. So the homepage is different. Open Babel, I know. Babel is okay. I think this is the correct one. Okay. Through this, you can able to convert the formats. Like how Word to document is something you're using different softwares, isn't it? Word to PDF converters. Likewise, you can use this for converting one to another. Okay. We have to check for Babelwin or open. Generally, most of the people try with Open Babel. Okay, let me take you to the today's session contents on special databases. Okay, I better close the WhatsApp. Yeah, so we can come across how long will the recordings be available after the end of the sessions? It will be available, but participants, it's a nice question from you. Actually, all these videos are presented, getting recorded in my personal Gmail ID. So the Gmail ID, I think the maximum account in the drive is 15 GB. Now it's already filled 15 GB. Okay, I suggest you to download the videos once in 2 or 3 days. Okay, at least like if you see the contents, okay, like I don't know, or else I have to transfer some kind of a videos to Praveen sir, but it will be there until you complete the course. We manage in such a way that until you complete the course, the recorded videos will be there. So, but if you want it in your system, even you can download it. Okay, whatever you feel right, you can make it out. Okay, but we say ensure that these videos will be there. Okay, up to the last day of the class, that is 6/9/24. Take care. Okay, because it's defaultly taking my Google Drive for storing the videos, and already it's full. Okay, before going to today's session, I can also give you some kind of information. Um, for there is a opportunity for you, like to get the jobs and internship in domain of artificial intelligence. This kind of a training is provided by Indian Institute of Technology, Mandi. Virtual, okay, in online you can participate. So senior faculties are going to teach you. Okay, whatever you learn artificial intelligence, you can deploy the concepts in biotechnology and try to utilize it. Okay, so of course this is a recent domain. Anyone can prosper, and even from the biotechnology domain, if you want to prosper, you can make it out. Okay, so this is the link. Okay, the link for free internship. Okay, and they can arrange jobs after successful internship from IIT Mandi. Okay, related to artificial intelligence. Okay, you try to link with the bioinformatics. Yeah, now let me take you to the today's session. So actually, if you see this databases, last session, last month, I was taking a certificate course on applications of artificial intelligence and machine learning for the treatment of cancer. Okay, so for the treatment, I mean, to develop tools and databases. Excuse me. Yeah. So, like for developing the cancer databases and replacing the tools. So, we have discussed on the last month. So if you see the cancer databases, we have exclusive databases for cancer, for this type of cancer. Okay. So there is a common databases also for working on cancer. So for example, if you want to work extensively on the cancer genome, because what is cancer actually? Abnormal cell proliferation. If you find an abnormal cell proliferation, definitely some genes are overexpressed and some genes are underexpressed. So you have to take care of both more overexpressed genes or more underexpressed genes. These are responsible for causing the cancer. If so, if you go to this URL, mycancergenome.org, you can able to find, you can specifically search for a disease. Now I search for hepatocellular carcinoma. You got list of diseases. If you click this plus, you get even the expression, how much expression AKT1 has expressed so in the cancer when compared to the normal patients. Okay, it depends on the types of cancer. May here if you get like 2%, might in breast cancer it might be 5%. So then if it can also be a significant. If it is above 20%, definitely it is a more significant gene for the cancer. Okay, plenty of genes, thousands of genes are responsible for a cancer, but you should try to find which is the more significant by checking the expression, like more upregulation or more downregulation. Sometimes more downregulation gives you a significant gene for gene therapy, okay, for the treatment of cancer. Okay, so this is a kind of a website, mycancergenome.org. If you want to specifically go to a carcinoma kind of a database, that is HCC database, we have this kind of an URL, predicted. So if you go through that, you can find this information of the genes. And there is also one exclusive database for hepatocellular carcinoma, oncotv.hcc. Okay, and if you see this database from ISCOR Delhi, okay, scientist Raghava has developed so many tools and databases for the biology purposes, especially he has created for even for cancer. He has created more tools and uh, for the cancer. So this is one such database actually for checking the biomarkers. What are the most significant biomarkers for the diseases? Okay, so he has included the wet lab experiments and he got the kind of a data and the data sets are deposited in this web server. And the common database, the large database like mycancergenome.org, this is also a common database. C Bioportal. Okay, like how you find NCBA. In NCBA, you can find GenBank, GenPept, Genome. Okay, so many databases, isn't it? Including for the literatures. Similarly, if you see for the cancer, if you want to work on cancer, okay, the best large database, okay, when compared to normal, the general databases. In what you find in NCBA, you just get a sequence for it in NCBA in GenBank, or you get a structure from MMDB. But how about the expression patterns? What are the different significant genes? What are the most expressed gene and how about in different regions? Okay, so you find different data set, one data set from the US people and another data set from the Asian US people, okay, Asian American people. So you find different data sets and even whatever you find different racial discrimination people. Okay, so if you find this exact what kind of people, like whether you're treating the Latina people or you're treating the Asian people in US or what. So through that, you can find the significant gene. Okay, so may in America for the breast cancer, BRCA1 might be the most significant gene. The same if it is an Asian in America, might be BRCA2 is the most significant gene. If so, the treatment will be different. Okay, the target is different, and obviously, a kind of drugs, whatever they prescribe, and whatever kind of therapies they clinicians prescribe will vary with each other. Okay, so one should have a knowledge. Okay, not to just get knowledge from a single data set. The multiple data sets are there, and even multiple mutations and what mutation is significant. So the cancer is, of course, also a hereditary one, isn't it? So through even through hereditary, you can able to find the cancer is spreading from the ancestors. So if so, what genes are responsible for the hereditary? How, like we have discussed, OMM is one of the important database, but still for more details on that, you can find in this database. Okay, so you can try initially like cbioportal.org on today. Okay, after my session, and you just type a cancer. Can be oral cancer. You can type breast cancer, something like that, and you find such a kind of significant data. If you scroll down, you can find more data. I just showed you only the one page. Nearly 5 to 6 pages are there. Okay. I also mentioned you on OMAM, isn't it? In OMAM, you have to search for a disease. You get a kind of a multiple entries. You can also find data sets and what are the significant genes and how how this disease is linked with other diseases. This might be of a viral infection, or you may get a kind of a I mean, the cancer is linked with also some viruses. There is a relationship between virus and cancer, or especially if you see cervical carcinoma, people have 99.9% infections. What infection? Human papillomavirus infections. Likewise, each and every cancer is also associated with some kind of a viral infections, especially you see the hepatocellular carcinoma, a primary liver cancer. These patients have 75, a minimum 75% of hepatitis infections. Can be hepatitis B or hepatitis C. Okay, so all these links, okay, so whether it's a hereditary or it's a kind of an, so most of the hereditary related you can find in OMA, okay. But in CBio portal, you can find even the infectious informations, etc. Okay, so what are the various causes? All these informations you can find in this common portal. There is also one more common portal called Genome Data Commons. Genome Data Commons data portal, where you can find even the clinicians' data. I mean, the patient's data deposited by the clinicians. Okay, you can find what are the significant genes they found on the types of cancer and how many mutations. Generally, this database is maintained by NIH, like how NIH is taking care of NCBI. Okay, that is this is not from NCBI portal. This is a different portal, but this is maintained by NIH, like how NCBI is maintained. Okay, likewise, the NCBI is maintaining so many portals. Okay, for the types of diseases. Okay, so if you want to work on this, you have to simply type this URL, and we have to type the name of cancer or a gene. Obviously, get plenty of information on it. Yeah, even in NCPA, you have GEO profiles. Okay, I just expand you on GEO. There is also another database called GEO profiles. Simply you have to type a disease name and you check it. Not just the cancer, you type even arthritis, autoimmune disease like psoriasis, whatever you type, okay, you get information of the data sets. That is the data sets or nothing but uh, information from multiple patients from the same hospital. Most probably they take from the same hospital. So like a 2 months data or 3 months data. So one data set can contain 50 populations data. So another data set may contain 75 population data. It depends on how much duration they're taken, how much patients are approached during that duration. Okay. Some information I can find related to that. So obviously you can find what is the significant gene, all this information. Gene cards. I think yesterday I explained you like this is a very largest database and fully annotated database like for protein in UniProt. If anyone want to know about the protein information, I suggest you to go for UniProt initially to learn about the protein. Similarly, if you want to learn about the gene, I suggest you to go for the gene cards. This is a human gene. This is see here, human gene database. This no you can't find for the mice or rats. So working on one of the clinicians can utilize this kind of information. Of course, even those who are working on human diseases. And the cancer proteome atlas. The next database is the cancer proteome atlas. Like if proteins are generally connected in a network, multiple proteins are connected in a network. If you're focusing on a particular gene, for example, if you're focusing as a most significant gene as PRCA1 for breast cancer, okay, what are the other proteins interacting with PRCA1 that also are considered as important targets? Okay, that also will be considered as important targets, and which is most influencing gene, which is most important regulating gene, which is giving, it can be an overexpressed or underexpressed. Even that kind of gene will be considered as one of the important target after PRCO. Okay, so one should understand about the proteome then. The proteome or interconnection of proteins in the network. So you can find for almost all the kind of diseases. Okay, whatever diseases they have deposited, you can find in the form of a data set. You can select it and even you can choose the type of network to find the information of that. For this, simply you have to type TCPA, the Cancer Proteome Atlas, portal.org. Okay. And similarly, you can find open targets. If you want to get some targets information, you can go to this platform. The most important database is the Cancer Genome Atlas, the Cancer Proteome Atlas, whatever I showed, CBio portal, mycancergenome.org. So these are common portals, but we also have exclusive portals for specific diseases. I think egg pathway I've discussed on yesterday. This is a JPR, that is gene expression server, like gene expression profiles for the cancers, almost all the types of. If you see the mouth, okay, so in mouth itself, we have mouth cancer separately, tongue cancer separately, teeth cancer, tooth cancer, okay, oral cancer is the common one. So each and every organ is can get a cancer. If so, okay, so what is the gene expression for a specific organ cancer? All this information you can look through this. Okay, so not only a single gene, multiple gene interconnected with it. Even you can get the expression patterns there. And enrich server for if you want to get a network and if you want to enrich the genes, then you can use this server. So before, uh, before going to these kind of a server, one should know how the protein networks look in the server and how the gene networks look in the server. So now let me take you to the website. Okay, I can Mala cards, path cards, Mala cards. I didn't hear path cards. You have to check. I think you have an idea on that. It looks like something different. Mala cards. Okay. But many people try to use the gene cards. Okay. So I have to look through all this. Now let me take you to the some secondary databases. Okay. Like SCOP. Okay. SCOP is structural classification of proteins. This comes under secondary databases, second or derived databases. So inside you can give an identifier, or if you know about a protein, give a protein name. Okay. If you know even UniProt ID specifically or PDB ID, even that can be given as a identifiers. Okay, as a keywords. So initially typed as a insulin. So for the insulin related proteins are shown here. So nearly 1,586 folds, superfamilies, families are shown. You can give any protein and you can look through that. Okay, so I just type insulin. So what are the insulin related proteins? Insulin, like you can find this much entries. Okay, it's slowly loading. Not at you didn't only one folder. Okay, only one fold I got. Okay, so the one fold you get some 8 entries here. So for each and every species you got one. Okay, Homo sapiens, Vostoris, Suscrofa. For insulin, like you have only one kind of a domain. Okay, I mean only one kind of entry. So to get inside, we can find this information. If you click this, it take you to the uniprot of insulin. Like if you click this, it take you to the PDB. PDB.org is different, and RCSP PDB is different. So you have two kind of entries. And ID is uniprot ID, and of course this is a PDB ID. Okay. So now we can see the this kind of a domain which is coming in the scopus. I mean scope. So this is this information you can find in scope. Okay, so the SCOP is one such a domain database. You know what is domain? Domain is a part of a protein which gives a function or a structure for a protein. The insulin is a functional protein. It regulates the glucose level, isn't it? That is the biological activity of the insulin. If so, to regulate the glucose level, what is the important part? What is the important domain? So these domains can be found from this SCOP database. Of course, the SCOP is present in EBI, European Bioinformatics Institute, maintained by UK. And there is also one more database, secondary database called CAT. SCOP, CAT, what else? Prince, PFM. Okay, these are some of the derived databases. You can find domains. Yeah, okay. Now let me take you to the illustration of CAT database. This is the second database. We call this as classification, architecture, topology, homology. Classification, architecture, topology, homology. Through that, the proteins are classified. I mean, the domains or proteins are classified through domains. Okay. Now let me take you to the human insulin. I have already taken to the UniProt and just copied a insulin. Okay, insulin FASTA format. I simply paste the FASTA format of the insulin. I believe I already explained you how to take a FASTA format. Either go to the NCBI GenPep for downloading the FASTA format or go to the UniProt. Okay, I've gone to the UniProt and fetched a FASTA format. Simply I pasted in the window and click search. If you click search, okay, you get some kind of a matches. So this is CAT structural domains. This is CAT functional families. For CAT structural domains, we initially got an entries 10 matches are found. But related to functional families, you can find 6 matches. Whether you want domains or family matches. If you want family matches, click here. So you get some information on that. Okay, you get information on the family matches. So one can get inside and also see how much is matching. So whatever is shown in the red color, okay, is matching. So the remaining gray color is not matching. So I can get this for this F3D structure if you click inside. Okay, the CATDB is taking you to the domain structure. If you want to focus on a small part of a protein and not an entire protein, then you can use this kind of a database. So what is the necessity to take only a part of a protein structure? Because if you go for simulation, there is one computational aided drug design technique called simulation, molecular dynamic simulation, where if you want to check the efficacy of a drug for a particular target, okay, there is a no need, it's not a mandatory that you have to use an entire insulin protein as a target. I mean, not an insulin, even whatever protein target you take, there is no need to take an entire protein. Even the functional part, like how I explained you for insulin, if functional part is more important, then that part is already you have a structure, so you can use it. So you can reduce the time in MD simulation. If you take an entire protein, if it takes 3 days for MD simulation, if you take such a kind of a domain structure, the important functional part of a structure, it may take 1 day for complete. It depends. So this is a small protein, but if you take spike glycoprotein of SARS-CoV-2, you know the size of the spike glycoprotein? The insulin size, the insulin ligand size is just 110 amino acid. Okay, the human insulin ligand protein size is just 110 amino acid. But if you see the spike glycoprotein of SARS-CoV-2, the size is 1333, very large. When you try to develop a drugs for the SARS-CoV-2, especially keeping spike glycoprotein as a target, it's very challenging to work on MD simulation because it takes much time. It takes 2 weeks or 3 weeks to complete the jobs based on the configuration. So better you can focus on the uh, domain, the functional domain. I mean, what is the most important part? If you see the important part of the spike glycoprotein out of 300,333, you find only 13 binding sites. The region might be up to 50 or 60 amino acids, but The most important binding sites are just 13, 1, 3. So you have to take only the region specific. You take a 3D structure like. Okay, so for this cat can help you, okay, to find the domains. Or even if you can able to find the same in Uniprot, no problem. If not, you can come to this kind of a database. Okay. So now I illustrated even the secondary or derived databases. Okay. Now I can take you to the protein interaction database. For protein interaction database, the first thing I'm going to illustrate is string DB. The string is generally functional protein association networks. Okay, protein interacting with another proteins in network can be visualized in string. Okay, in this string, if you see the data, nearly 12,535 organisms data are available. And this much millions of proteins interaction you can find, and interactions are around 20 billion interactions. So greater than 20 billion interactions you can find. So to initially work with this kind of a protein-protein interaction networks, just get inside, okay, type a name of a protein. Okay, so I can give some significant protein like can be a BRCA1. Okay, I'm going to type a protein name as a BRCA1. And if you want to work on human, you can try with the Homo sapiens, you search like that and you get it, or else simply go for search. I just click yeah, I got a Homo sapiens now. Select it. Okay, escape. Some problems then. Select. BRCA1, Homo sapiens, select. Search. Now it just showed you the Homo sapiens BRCA1. But BR7 is also complex with few proteins like PABM2. So you got a second entry and another entry you got. But you are focusing on only BR7. So this is the first entry in default. Simply click continue. The human BR7 you can find here. Okay. If you click this BR7, you get the detailed information of the BR7. Okay. And identifiers. How about the 3D structures? PDB ID is mentioned here. Okay, so another PDB ID, how much identity. So you can find each and every PDB ID, or you can find even the alpha fold ID is like this, like a Uniprot ID. Okay, and how much identity. So get some structure information also and other details. So if this is your major protein, okay, if this is your major protein, so there are some interconnecting proteins in the hub. These are interacting proteins of VRC1 in the hub, in the network hub. Okay, nearly the first 10 proteins alone is shown. But if your target is more than is up to 50 proteins, then instead of 10 proteins, if you want to focus to find what are the other 40 proteins, okay, you can change the same in settings. In settings, instead of no more than 10 interactors, click no more than 50 interactors. If you want 100 proteins, give custom value and give us 100 value. Now, if you want to find just 50 interactors of BR7, I just selected that, okay, and simply click update. Now you can able to get 50 interacting proteins with BR7. Okay, so BR7 is this is what BR7? No, this is BR7. So this protein is interacting with 50 proteins. You can change the number and you can check, okay, what are the, this is of course the most important target, so you can find many interacting proteins. Okay, and if you want to see some other description, like if you want to download these networks, okay, these networks can be downloaded in the form of tabulated separated values. You know, the tab separated values can be opened in Excel, Microsoft Excel, or even if you are using a Linux, you can go for such a Star Office and a Star Excel, something like that, Open Excel. And the general best visualizer to find these networks is Cytoscape. The Cytoscape is the freeware, okay, used to visualize protein-protein networks, gene-gene networks, and metabolic pathways. All these things we can able to find it in cytoscape. Okay, the network related informations. So generally you have to click this uh, download hyperlink. And if you see this kind of BR7 protein, okay, if you want, if you are focusing on specifically human breast cancer, okay, and the two specific pathway, okay, you can able to select a pathways, etc., as an advanced filter and select the most important genes. We got multiple entries. Server load is 34%. I think the server is oh okay. Now we can see this information from analysis. In analysis, okay, whether if you are focusing on breast cancer and also linked with ovarian cancer, think like you are linked with both breast and ovarian cancer, hereditary based, then you select this. Whatever you have selected is shown in red color. Okay, out of 50 proteins in the network, you can find only 1, 2, 3, 4, 5, 6. Only these 6 proteins are connecting breast and ovarian cancer. Okay, so likewise, if you want to have a second filter, okay, like the filter is something related to a lymphoblastic leukemias. Okay, T cell acute lymphoblastic leukemias. You select this as a second filter. Okay, so which is like some patients have a kind of a metastasis and it can distribute, and might be this is the major reason for that. Okay, if so, I'm taking a second advanced filter like a kind of a T cell based acute lymphoblastic leukemias, okay, which is shown in blue color. So now whatever out of 6 proteins, only 2 proteins are shown as the major, and that too, this is something new. Only one protein you can find. The BRCA2 is linked with the cervical and as well as the T cell lymphocyte. So this is the most interacting protein for a specific patient. So if you are working as a BRCA1 as one of the important target, you should also give importance to the BRCA2. Okay. Now we got it. This is how even you can able to find the important proteins in the network through these filters. Even you can do the filter through the pathways. Okay, if you are proficient in working on a cancer or even other diseases, not only cancer, you can get any diseases information from the protein-protein interaction networks. Okay, even during the metabolic pathways, these kind of proteins also can find in this protein-protein. Not on the disease pathways, even metabolic pathways, you can find in networks in string-tb.org. Okay, I think now we got some information on the string-tb.org. And if you want to find the sources, what are the sources they have used to get these kind of protein-protein interaction networks, you can find the same in viewers. In viewers, you can find experiments. Okay, if you click these experiments, you can find what type of experiments they have used in different. You can find different multiple data sets. So whatever protein network they have established is not from a single experiment, but these are done in multiple experiments. Through the multiple data sets, they formed a network in the database. So if you say this, okay, one such data set, you can find this much, how much? 125 plus around 25, 150 proteins. 150 proteins are linked with the PRCA. Here, if you see in another kind of experiment that is done through biotin identification assay, proximity dependent biotin identification assay. This is the second assay they have done. So here they have found 255 plus some 20. So nearly 275 interacting proteins. So this might be some 5 patients, this might be 10 patients in a sample. Okay, so through that, whatever data, so we also find one more experiment, a different experiment. They have used tandem affinity purification assay, co-immunoprecipitation assay. So you can find immunoinformatics assays, like I mean immunotechnology assays. Immunotechnology assays we can able to find a different assays, and you can find even what are the genes identified as the most interacting genes for a specific gene you have selected. That is, in our case, BRCA1. Okay. So likewise, you have much details. One can go to this website and simply search with a gene and find multiple information on this. Like however we find a stringed DB, if you want to find uh, the drug interacting proteins, you can go for stitch database. The stitch database, I have to check whether it's working. No, presently it's not working. Yes, working now. Sometimes the stitch DB gets hanged. Okay, so then the server is on under maintenance. Now, no problem. The server is working for us. Okay, the string DB is different and stitch DB is different. How it's different? That is actually protein-protein interaction networks, but this is chemical protein interaction networks. So the drug-protein interaction is very important. Okay, for the disease treatment, for checking the efficacy of a drug in treatment of disease. One should know how much time the drug can bind with a protein. Okay, which we can find in simulation. Okay, so for this, you can also come to know about this gives some kind of an information, a basic information on how the drug binding with the proteins. For example, if I click search, okay, I'm going to type a kind of a compound. Okay, this is a plant compound with aferin. This is a principal component of ashwagandha with somniferum used for treatment of 13 types of cancer. Okay, I simply type a compound name, which is I use like a APA, active pharmaceutical ingredient, a form of a drug for treatment of cancer. And so checking for that compound. You get an information if you give as a keyword, or even can give a protein name as a keyword. Can give either a drug or you can give a protein name as a keyword, and you can find drug and protein in the form of a networks. Okay, this is a kind of a network database. Like this, I think even on yesterday I explained you about KIG, Character Encyclopedia of Genes and Genomes. I told you to either give us a protein as a input or give a disease as an input to get a kind of interconnection of proteins in the database. Okay, not only the disease, also the proteins and metabolic pathways also can find in cake. Okay, I think it's taking some time. It will be generating the result soon, but meanwhile, I can go for the other informations. Yeah, just after this one, I can go to the enricher. Okay, once I told you what is string TV, how the protein network look like, okay, and what are the different functional enrichments available. I just taken some kind of tissue expression, something like that. But we also have your pathways like cake pathway, reactive wiki pathways, etc. These are advanced filters. You can select any number of advanced filters and try to find which is the best interacting gene with your target protein. Okay. Here in my case, I'm working on hepatocellular carcinoma, so I have selected liver from the tissue expression. So you get only whatever is selected as liver is shown in the red color. So if this is my main target, heat shock protein, 5 proteins I can able to find in the network. But when I selected for the second advanced filter, other than liver organ, I have also selected, I want to check only the hepatocellular carcinoma. That is the influence of heat shock protein on hepatocellular carcinoma. I can find many blue colors. But if you see the liver with hepatocellular carcinoma, where I can able to find 1, 2, 3. These three are the most significant genes. So one has to target these three important genes if you are working on a specific heat shock protein of an organ liver. You're targeting the hepatocellular carcinoma as the major cancer disease. Okay, so whatever I got, I showed you here as the important protein. So one can able to find the interacting proteins like that by functional enrichments, which is available in the string itself. The string is a database. Okay, And similarly, compound interactions in stitch. When you search for vithafirin, you get an output like this. Not only you can able to get a drug that is a API interacting with proteins, but also you get how a drug is linked with the other drugs also. Okay, if other drugs are given or used as an inhibitor for few targets, okay, whether you can find a influence on that. That is like if you go to a doctor and if you tell I'm suffering from cold and cough, whether he'll be giving a single tablet for cure? No, isn't it? Give for cold, he give a separate, and for cough, he gives separate, and based on throat infection or your, how you feel, whether you feel easy or you find other complications, then he give another tablet for that. Okay, an anti-acid tablet, some four tablets. So you get these tablets. And so one should also know about the side effects of the other drugs, which can be given along with the usual drugs. So even the other drugs kind of interaction also is shown here in the stitch. But my search is only for the vitafrini, but you can also get these information. Okay, and here also you can use functional enrichments. Through functional enrichment, I can come to know which are the important protein targets. Only the proteins alone are selected and shown. Okay, I've used a functional enrichment to find here. And even this kind of a string or stitched AB, okay, these databases, if you want to see it in a software like Discovery Studio Visualizer, the Discovery Studio Visualizer you can't able to use for visualizing protein networks. Okay, but what is the exact software you can use? You can use Cytoscape. However, you find a view in Discovery Studio Visualizer for protein networks, a very nice view you can able to find in Cytoscape. So, here I didn't show that, but I can show you in the next sessions how you can find this network in the Cytoscape. If you have installed a recent version of uh, Cytoscape from the stitch, if you click the send network to Cytoscape, it directly takes you to the Cytoscape software. Is network. No need to download the Excel sheet in TSV and again open in Cytoscape. There is a direct transfer of data to the Cytoscape software. Okay, so what you have learned on today, majorly you have learned on various cancer databases, a specific cancer databases. We have discussed about derived databases like what is CAT, what is COP, and how to go through that. Okay, databases and how to search the data. Okay, so this information we have seen. CAT, I told you already, class architecture, topology, and homology, and SCOP is structural classification of proteins. The proteins are classified based on domains, isn't it, in the form of families. So whatever classification they made, here you find uh, seven classes of classification of proteins. Okay, this is the major and this is submajor. And like this, we find seven levels of classifications. Okay, here you find four levels of classification: class architecture, topology, and homology. And one should also have a knowledge on sequin and pancake. Sequin and pancake are the deposition tools for sequences, can be protein, mRNA, etc. If you want to deposit any sequences, you can use either sequin or pancake. Generally, we use pancake. Okay, so it's a sequence submission tool, and it's a web based. Okay, and here you see simple sequences are submitted using pancake. Some complex sequences are submitted using sequin, where you have to first download and install the sequin, and then you have to import the sequences through that. So most of the sequences we try with the pancake. Okay, complex alone we try with the sequin. So these are some of the data, the data deposition tools present in NCBI, National Center for Biotechnology Information Web Resource. And if you want to see the genome specific databases, thousands of databases are available. The popular one we discussed about NCBI genome, TAGR, KIT, etc. So we can also find some more information about genome in www.genome.gov. In NCBA, we have NCBA Genome, but there is also a website called genome.gov. You can find more genomes information on this. So nowadays, if the clinicians want to diagnose a disease, they're not focusing on to check with a single genome, but they're trying with 1,000 genomes. We have 1,000 genome projects. Okay, so before 5 years, you can able to find this information, but now at present, those are 1,000 genome projects are superseded by 1 lakh genome projects, and maybe within 3 to 4 years, we may get 10 lakh genome projects. So what I mean, 1 lakh genome project, it means that 1 lakh patient genomes are taken for comparison. Okay, if you're focusing on a syndrome and you can't able to check with a normal healthy patient of one genome, even if you check with 1,000 genomes, you can't able to find a inference. You can't find a conclusion for a RAND syndrome. If you check with the 1 lakh, you may get an clue to find the appropriate gene which causes the syndrome. If you try with 10 lakhs, you get more precision information about the genes. Okay. So now at present, we have 1 lakh genome project for humans. If you are working on humans, it's easier for us. And even I think we have 1 lakh genome project for rice, Oryza sativa. We have black rice, other than white rice, we have black rice, red rice, so many rice types of rices are there, isn't it? Okay, so for this rice, we have even 1 lakh genome information. Okay, so you can use for plant biotechnology or animal biotechnology using this genome website, genome databases. So, but whatever I showed here, NHRI, this is National Human Genome Research Institute. Okay, so human genomes information you can find from genome.co. And for plant genome, okay, the National Institute of Plant Genome Research, NIPGR, you can go to this URL. So many plants, okay, like if you, now I was explaining something about ashwagandha. The ashwagandha comes under Solanaceae family. You know what you mean by Solanaceae? Solanaceae is a tomato family. Ashwagandha berries looks like a tomatoes. Okay, so there are many plants coming in the tomato family, which we call that as a Solanaceae. Either you find a Solanaceae genome separately, or even for a specific tomato or ashwagandha, also we have a separate genome website. Not only you can find entire thing in a common website, even for a family of proteins, or even for a specific species, also we have. Uh, I think it takes more time for Vitafrin. Some problem is that, but I already showed you. If you type a Vitafrin, what kind of output you get in PowerPoint. Okay, let me close this. So if you want to go to the Solanaceae, okay, Solanaceae, yeah, the Solanaceae genome, simply type like this, okay, you get, yeah, salt genomics. Even if you want to specifically go for a tomato, give a tomato genome, you get a different name. This is for Solanaceae, salt genomics. Now I'm going to type the same for tomato. You have, likewise, you can find metagenome. So tomato genome consortium. This is saltgenomics.net. This is also not right, but we also have an exclusive database for tomato. I think this might be tomato genome sequencing, cdi-genomics.com. We have for species for a specific thing. So 100 tomato genes. Okay, like 100 tomato genes. I was explaining about 1,000 genome project, isn't it? Simply type like 1,000 genome project. So take you to the UPSC International Genome, 1,000 genomes. Not only can find human, you can find for uh, the plants and the mice, rat, etc. Okay, 1,000 genome projects are established for many species. You can search for a specific thing in 1,000. Not only have 1,000, I was mentioning you about 1 lakh genome, isn't it? You can check it out. Yes, this is done by Genomics in Lab. 1 lakh genome project. Okay, so get more precise information if you are searching for a specific disease, a rare disease syndromes, etc. in this website. So these are actually the genomes are established in 2001 to the final draft of the human genome was published in 2003. But after genomes, many scientists focused on establishing proteomes. Okay. So recently we are working on protein-protein interactions. Even myself, I'm working in protein bioinformatics lab. So I'm focusing on this kind of a proteomes. These are outdated genomes, but still these are getting updated by increasing the size. 1,000 genomes, 1 lakh genomes, etc. Okay. So this is something about what's happening in the world. Okay, on a day-to-day research. Okay, I have completed the kind of information to be delivered on today. Anyone have any doubts? Want a cytoscape demo? You can able to find much information on the YouTube. But during the mini project discussion, I keep it in mind and try to explain you. And also other softwares. Okay, I can teach you during that time. Any other doubts you have? How protein-protein interaction is useful? What type of information? So this you asked at 7:45, but now I think at 8 o'clock we got some information, isn't it? Like if you have two different courses, you can select the two different courses, two different, that is functional enrichment, and you can get most important significant thing. I think this I have explained, string and stitch. String is just protein-protein. This is chemical protein interaction. So what computer programming are important for bioinfo? Computer programming, for R programming is majorly used for handling gene expression data sets. Okay, if you want to use a bioconductor software, microarray gene expression, RT-PCR gene expression, you can go for R programming. If you see in Java, we have exclusive data sets to handle the sequence and structures. So instead of Java, you can go for BioJava. If you see the Perl, you find many packages are majorly used to handle the sequences and structures. So whatever Perl you use, default it is a BioPerl. Okay, these are specific languages for handling bioinformatics data, unlike C, C++, etc. Sir, how can we upload a DNA or a protein sequence in NCBI? Simply, you have to go through the portals. Just search in Google like a Bankit, and you just try to deposit it, obtain from a factory or warehouse. Just in Google, you search for Bankit. Sir, will be a learning molecular dynamics simulations. How come? To understand our syllabus, dynamic simulation, I should have a workstation. Okay, the workstation should be a minimum. The nowadays the price of the workstation ranges from 2.5 lakhs to 12.5 lakhs up to 20 lakhs. I should have a workstation to explain about MD simulation. And the two for a demonstration, it takes nearly 2 to 3 days. Full 2 to 3 days. What is the significance of E value? Till now, I didn't explain you all this. So when we come to the blast, I can explain you. Okay, thank you very much. I think I've answered for your queries. Okay, but whatever most of the questions you asked, okay, I have yet to cover the syllabus. If and if I cover, then I can answer for all this. Okay, we don't have the download permission. No, I don't think so. It's restricted, is it? You can only view the video, but you will need to give permissions. Actually, I'm not. I don't have rights to. I mean, it is storing in my drive, but I can't able to give the permission. Okay, it's with the coordinator. You have to contact with him. I think you can visualize then. Only from you I can come to know. Okay, okay, I understand. And sir, how can we download the videos? Uh, See again, same question. You have to check only the coordinator. Okay. So thank you. Okay. Thank you very much.